Hello.
Hello.
How are you?
Pretty good. Yeah.
Yeah. So it looks like we've got Hussain's here. Hello, Hussain. And Vahid's here.
Hello, nice to meet you all.
Thanks.
And Saurav.
I'm the new one.
Yeah. Saurav, would you like to introduce yourself?
So, my name is Saurav Kumar Roy, and I'm from India.
Okay. What are you interested in? What are your interests?
Actually, I need this field, so I'm able to learn.
Yeah. Well, glad to see you here. Welcome. Looks like Mehul is here as well. Welcome.
Hi, everyone.
Hi.
Hi, Bradley.
Hi. All right. And Hussain is here. I don't know if you've ever introduced yourself to the larger group.
No, this is my first time. This is my first time hopping on board with DevoWorm.
Okay.
Yeah. Hi, all. Nice to meet you. My name is Hussain. I've done some previous research under Bradley before, and I'm hoping to get on board with the DevoWorm project. I just finished my graduate studies at the University of Toronto in medical sciences, where I did computational neuroscience research. Moved back here to the United States while I'm getting involved with a lot of remote positions across tech, science, research, machine learning, and happy to extend a set of hands to help with DevoWorm.
Good. Thanks. Mehul, could you introduce yourself?
Yeah, sure. Hi, everyone. So I am a MS research student at IIIT Hyderabad, India. Currently, I work in medical image analysis, to be specific, brain imaging. So my work is applying graph neural networks in the brain data. So I've submitted a paper applying hypergraphs on it, and I'm currently working on directed and signed graphs. So
Good.
I'm looking forward to learn from everyone here.
Yeah. Morgan is here, and so is Jesse. Morgan actually does a lot of stuff with neuroimaging, so he's interested in that area. But yeah,
that's interesting.
Yeah. So welcome everyone. So we have a bunch of things to talk about today. So let's get started. Unless anyone wanted to give an update on their things that they're doing.
I don't know. I'm doing the math of tensegrity right now. Lots of fun and games.
All right.
Linear algebra.
All right. Yeah.
Yeah.
And how about Vahid? Would you like to give an update?
Yeah. As you know, I've been working on the developing C. elegans. Actually, the models that are working on developing C. elegans. Fortunately, I could find one study that recently is going to publish maybe in the future, because in the paper is somehow the preprint, the paper that I posted in the group as well, is a preprint which has no data for now. They have mentioned that in the future, when they are going to publish the paper, they will release the code and maybe the data. But now they haven't included the data in the preprint paper. So that was something. Hoping maybe in the future we could have some data, and it's specifically they have worked on L1 worm. They have some data regarding the movement of C. elegans during its L1 development cycle. And I also found one more publication from 2011, I guess, but they are not that much data oriented. They just have some observation on some developments during actually some movement or locomotion of C. elegans during different larval states. But the one that I found recently, maybe they are working. They have sort of recorded data for the L1 larval stage, the movement of the C. elegans. So that would be a little interesting for our modeling approach.
Yeah, I saw that in the Slack, that paper. Is that the same one, or?
Yeah.
Yeah, I'll take a look at that later. So yeah, thank you. That's good that you're finding stuff like that. There's always stuff that I've missed or don't know about, and I see it come up, and I'm like, oh. So yeah, people are definitely using C. elegans as sort of like a first place to go if you're trying to quantify things like development or so. All right, so yeah, let's get started then. Let me share my screen, and I'll have a couple things to say sort of in brief here. The first thing I wanted to point people to is that there's this new podcast. If you're new to OpenWorm, this is useful, I think, to listen to because it talks a lot. This is Steve Larson who's giving the interview here. He's the CEO of MetaCell, but he's also one of the founders of the OpenWorm Foundation. So he talks about, this is on Spotify, he talks about the OpenWorm project, the OpenWorm Foundation, and the sort of, I don't know, he gets a little bit into the history, but he also talks about, what you can do with data from C. elegans. So, this is a very general audience type podcast. I don't know, it's like 34 minutes, so it's not too long. But basically, the whole sort of hope when OpenWorm was started was that you would be able to use C. elegans as a means to model an organism from like the cells to behavior and, have a good computational model so that you can simulate the worm. So this worm here is a microscopy image of a worm, but, we have a Docker container that you can download from the GitHub, the OpenWorm GitHub. And if you run that Docker container, and it takes a little bit of effort to do so, you can get a series of models to run that model the biophysics of movement, that model some aspects of the connectome, and some other simulations that are in there that will run, kind of like almost like a movie. And the idea is that you can take those models, extract like information from the simulation, use those parameters then to model C. elegans, and it should match the actual C. elegans worm. And so, there, I guess because C. elegans is actually a pretty easy target, it has a lot, for example, C. elegans has a tractable connectome, 302 cells. It has the stereotypical movements. So it can move, it basically moves, only has a couple modes of movement. Generally speaking, you can actually model those things. And we're going to get into the paper that Vahid put in the Slack later, so we'll talk about that. But basically, that's the hope. And we're not really that close yet, but as you can imagine, it's a pretty long-term project. So this is definitely something you should check out. It's on Spotify. It's Data Skeptic is the name of the podcast. And this is the episode on OpenWorm. Okay, yeah, so that might be of interest to people. Another thing is that every year for Darwin Day, which is February 12th, I do a blog post on the blog that I've been running for like 10 or 15 years now. It's actually, I think, been 16 years almost, where I pick a topic in biology or evolutionary biology, or in this case, biophysics, and I kind of go through some aspects of that topic. So this is called Charles Darwin Meets Rube Goldberg, A Tale of Biological Convolutedness. So I went to a generative AI engine, and I typed in the prompt, Charles Darwin Rube Goldberg Machine. And these are some of the images I got out. So I was glad that I could get this as an image because it didn't exist in Google images. So this is basically, this is supposed to be Charles Darwin, old Charles Darwin. And this is something they call Rube Goldberg Machine. So if you're not familiar with this, this is a there was a cartoonist at the turn of the last century named Rube Goldberg, and he did these cartoons where he drew these machines that were like these elaborate machines that would do some very simple thing in many steps. So it would be like, I want to build a machine to fluff my pillow. And he'd build a machine that had like 100 moving parts to it. And it was many, many more parts than you needed to actually do this. And so, it was supposed to be sort of absurd, right? There's an absurdity to it, and that you need this many steps. And at the time, of course, in society, people were building machines to improve your life. And so there were some pretty outlandish machines at the time that would do things that were sort of like, absurd. And so that was just a thing that he was lampooning. But these kind of machines are interesting because, it takes some skill to build them. You have essentially a machine that is over specified, I guess you could say. And the reason, there may be lessons in here for biology. So if we look at like a map of a metabolic network, it's very, very complex. And the question is does it need to be that complex to be optimal? Or does it exist like this Rube Goldberg machine where you have all these moving parts and maybe more parts than you need? And so this is another type of image that was generated. So in this post, I basically talk about this paper I wrote about, I think it was about 13 years ago now, the machinery of biocomplexity. And this is a paper where I use these models called biological Rube Goldberg machines, and I use them to model different processes and consider, if this is something that will explain sort of the evolution of things that you see in nature that maybe aren't optimal, or they're very, maybe needlessly complex to do something to perform a biological function. So I go through some of these possible biological Rube Goldberg machines and how they might work. So then this whole post is basically going through this paper, revisiting it. And I didn't realize I had given a talk on this a long time ago at a conference, and it was kind of, it was okay. I mean, it wasn't that wildly received, but it had I basically went through this and I got some feedback. So this current version represents the comments that I got. And so there are different ways you can set up these Rube Goldberg machines. You can set it up as a simple process model, and then you can do things like mutate that process model or co-opt parts of the process model, where you can invert parts of it and copy and do all these different operations. And you can end up with this kind of thing that's sort of a machine that's superfluous in a lot of ways. And so this is kind of the thing we're trying to do. You can also do this with a microfluidic model, where you change the shape of the channels and sort of the way that it goes. And this gives you this sort of complexity, something that looks like complexity. And that's so the lesson here is that, evolution, people always say that evolution is optimal. And I would disagree with that in two ways. One is that it doesn't always give you optimal things. It gives you things that are sort of fit for the environment. And so if organisms are always moving between environments, you can have suboptimal things going on. You can have suboptimal traits. The second is that you have sort of this, you have things that you have constraints that evolve. You have to evolve within certain constraints. And so you can't always tightly optimize things. And so there's this idea in the literature called constructive neutral evolution, or CNE. And this is something I cite here. And there's been a bunch of work on this in the last 15 years, where people have looked at neutral evolution, which is evolution without an adaptive imperative. And so this is something else that's relevant here. So yeah, this is go ahead.
I appreciate the analogy and the comparison. A Rube Goldberg machine, though, they are on some level trying to optimize themselves too by being as convoluted as possible.
Yeah.
Like, of course, the trajectory, the goal of, say, pouring coffee or kicking a ball isn't the goal. It's everything fascinating that occurs before then that leads up to it. But that's so in that sense, it is optimizing itself to be as flashy and intricate as possible. So yeah.
That's a
Yeah,
good point.
it is a neat idea. Like, what is the most complicated way you can pour water? What is the most complicated way you can do the final task before you get it done? So I think Rube Goldberg machines, as interesting as they are, they're more so into the more like say fascinating idea of say suboptimal functioning, in the sense that whatever is being done before the final step, which is the goal, like whether it's say pouring a cup of water, whatever is being done before then, for whatever reason, the system is doing such a poor job of integrating information as effectively as efficiently as possible, and as such, it resorts to doing 50 other tasks first before doing that goal. So it's almost like not so much like, I mean, you could say it's like trying to make it as complicated as possible, but more so it's I see it as more of an analogy or a symbol of inefficient processing. So, but yeah, I appreciate that. This is a really cool comparison. When I was in school, I made one that would like do a bunch of stuff and then strum a guitar at the end. It would like have a pick fly and it would strum a guitar.
Yeah.
And it's like, that's how you strum a guitar. And it had like 50 steps before that.
Yeah, I do talk a little bit about that, but I never really got to the point where, like really kind of thinking deeply about that. That's why I kind of brought it back up, because I'm kind of thinking about it and I was like, well, this game seems kind of unfinished as an idea.
Because in some level, it is optimizing a function of some kind, but it is optimizing a function in more so how complicated and convoluted you can be,
Yeah.
Not necessarily with respect to the specific final step. Yeah. So that's what I would see about this thing. So
Yeah, well, thanks for the comments. Yeah. Yeah, so that's the thing for Darwin Day. So that's on the Synthetic Daisies blog, which is syntheticdaisies.blogspot.com, and it's the most recent post there. So this is the paper that Vahid talked about. This is it's a preprint from bioRxiv, and this was posted last summer. So I think this is the latest version. And yeah, I don't know if they published it anywhere since, but this is yeah. Oh, you wanted to say something, Vahid?
I was just saying that I've been looking for the published version because I found also I actually sent the link for some maybe the PubMed Central, but it has also been mentioned that it has not yet been peer reviewed by a journal, so it's not the published version yet. And at the end of the paper, they have mentioned that they did not publish any code or data anywhere until they are not going also to publish until they maybe formally publish the paper somewhere.
Yeah, it sounds like what I thought. Well, it was on PubMed, but the PubMed stuff, they have preprints on there now, so that's where it pointed. But yeah, this is a quantitative analysis of locomotor patterns in developing C. elegans. So this is where they're looking at locomotion in the developing C. elegans, and by developing they mean the larval stages.
I also have some previews on this paper and another paper, which is a link. Actually, it has been mentioned on L1. If you would like, after your introduction, I could connect this paper to another one that is actually somehow is describing how different neuron types in L1 and an adult C. elegans is actually working together to make some locomotion, both swimming and crawling.
Okay. Yeah, let me go over the abstract and then you can do that.
Sure.
So yeah, so the so this is let's go over the abstract. Adult animals display robust locomotion, yet the timeline and mechanisms of how juvenile animals acquire coordinated movements and how these movements evolve during development are not well understood. So we know like the adult movements, we have those characterized And in fact, in OpenWorm, it's one of the main imperatives of the project is to get, they have this movement database where they've gotten a bunch of microscopy images. So, you'll have C. elegans in a culture dish, and you can take an image of them crawling on this gel that that we usually plate them on. And so you get these, movements that they make towards food or whatever. Sometimes they're quiescent, meaning they stay still, but sometimes they move around that space and they try to, do things like interact with each other and eat food and things like that. So, but the thing is, we don't know because these larval stages are a bit different from the adult stages. They haven't quite gotten all of their cells yet. In the sense that there's some post-embryonic cell differentiation, and so we expect maybe some changes in both the connectome, which is people are working on that, and then the movement output. So that's where they're going with this. Recent advances in quantitative behavioral analyses have paved the way for investigating complex natural behaviors like locomotion. So, you can study locomotion just by simply letting a C. elegans crawl across the gel and do their thing, and you can just record it and then track it using tracking algorithms. In this study, we tracked the swimming and crawling behaviors of the nematode C. elegans. So they have very exquisite swimming behaviors, very stereotyped, but also very good. They're very good at swimming in like liquids. Like if there's a thin liquid layer that they have to traverse, they can do that easily. But also crawling, which is just kind of moving like a worm. And then they looked at this from post-embryonic development, which is there are these larval stages through adulthood. So they did this principal component analysis, and in this paper they use an approach called the eigenworm approach, which is something that OpenWorm is very interested in. It's a common approach in C. elegans movement. It's basically using principal component analysis, getting some eigenvalues, and then that's like characterizes natural modes of locomotion. So they call them eigenworms because they're these eigenvalue representations of movement. The principal component analysis revealed that adult C. elegans swimming is low dimensional, suggesting that a small number of distinct postures or eigenworms account for most of the variance in the body shapes that constitute swimming behavior. So they don't have a lot of different distinct types of movement. When they move, they use very stereotypical movements. So they might undulate back and forth at a certain frequency, and they might have 3 different modes of doing that, and that's about it. So they don't do these individualistic sort of adaptive movements. They're very stereotyped.
Do they know which part of the C. elegans brain is controlling the movements?
Yeah, so there's a movement circuit, and it has I think about 6 or 7 neurons in it, and they have one mode of that for forward movement and one for backward movement. So it's if it's going forward, there's certain neurons that are activated, and then going backwards, another subset is activated. But I think some of both movements use some of the same neurons, but they actually have it well mapped out, and there are other papers that talk about that. But yeah. Then our analysis revealed that swimming and crawling are distinct gaits in adult animals. So actually, in the larval development, they do these movements that are very much the same. They're very low dimensional. In adulthood, they kind of diversify their behavior. They have these very distinct movements for each type of behavior. So they're really refining that. And the explanation for that might be that those circuits are specializing over larval development. So they start with this L1 larvae, which is the first larval stage, right after they hatch from the egg. And then, you have up to L4, which is the final larval stage, and then adulthood. So throughout the larval developmental period, this circuit is sort of being refined, and then it specializes into these different movements. Remarkably, go ahead.
Hey Bradley, do you know? I suppose just in general, but also specifically for these sorts of studies of eigenworm space, Do you know in general how much of this can be extended or generalized to other species, similar species, say it's like C. inobita, C. inobita, like not just C. elegans?
Yeah. Yeah, well, I mean, they use this type of thing. There are a lot of species of stereotypical movements. So, like, not just like worms, but like other organisms where they're, you can track their movement. So, yeah, I don't know what like the stated. I know they do the eigenworm stuff and C. elegans, and you can use like that basic approach. I think in other organisms, like marine invertebrates and other worms, and I think in Drosophila as well. But I'm not sure what the literature looks like on that. Yeah, I think the idea here is that if you have like things that you suspect are stereotypical movements, you can like just do the analysis and it'll show that you can maybe use those to characterize the movements. So remarkably, young L1 larvae are capable of producing the postural shapes for swimming and crawling seen in adults. So they have these behaviors that you see in adults, despite frequent instances of uncoordinated body movements. So their movements aren't really all totally coordinated yet. We talked about how, in a previous meeting, how, in the embryo, you start out with these muscle twitches, which are when the muscles get kind of, before they even get connected to the connectome and they're differentiating the muscle, they twitch sort of autonomously. And then as they connect to the connectome, they kind of coordinate their movements. And so the L1 has this kind of carryover of uncoordinated movement, but they actually do have these behaviors that are, you can see them in L1, but they're not specialized yet. So in contrast, late L1 larvae exhibit robust coordination of locomotion. So this is where this L1 stage is where this is really beginning to specialize. While many neurons crucial for adult locomotion are still under development. So you have this core of like 7 or 8 neurons, and then other neurons are like modulating this circuit. And so that's what's maybe going on in L1 is that modulation is beginning to be put into place, or at least, learned, or, whatever. In conclusion, this study establishes a comprehensive quantitative behavioral framework for understanding the neural basis of locomotor development. So that's the abstract. Now, Vahid, if you wanted to talk more about the background, or I don't know what you had to share.
As you described, this paper is actually, as we said, is a preprint still. The main topics, as you covered, are first of all how they actually quantitized the movement of the worm. They decided to actually differentiate 11 parts of the worm into different movement shape, which are more like 4 different gestures that every 2 of them has been decided to be categorized as one type of locomotion, which is swimming and crawling. They have put this together to A1 and A2, which are the movement with these angles, to swimming, and these 2, A1 and A2, to crawling. So in that way, they somehow covered 97% of the movements using these 4 different gestures and angles. But what is interesting for us is actually these 2 methods that they have and works that they have done. They try to put different larval stages into the how the worm is moving. Young L1, which is after hatching, the late L1 after 9 hours of its life, L2, which is around 17 hours, L3 around 30 hours, and L4 and so on, which is from 38 hours to maybe 20 to 24 days of living of the C. elegans, which after L4, the size of the worm is usually around 1 millimeter. But they observe that all these larval stages usually generate some similar swimming and crawling patterns, but the young L1 C. elegans cannot reliably maintain the swimming rhythms. They also found that the young L1 C. elegans they have some type of immature crawling postures and locomotion. As you can see here, the blue lines describes the actually crawling postures of the young L1. So they concluded that during L1, especially the nervous system of the C. elegans is not yet mature enough to generate some sustained rhythms for swimming. This is the main actually discoveries they have done, but as we mentioned, they are going to put some link over here. That's the code which is usually interesting for us in Python is going to be published. So waiting for the code and data. But what Susan was mentioned could be maybe found in a paper like this, the elegans locomotion, small circuits, complex functions. In this paper, it's a little old, for 2011, I guess. But during this work, they have similar to many other papers like this, they have tried to somehow differentiate dorsal and ventral muscles that the C. elegans have, and some specifically those cells and interneurons and neurons that are somehow interesting for the locomotion of C. elegans. We can, if we have these 6 interneurons and these other 6 ones which are connected to muscle cells, you can somehow figure out how what is the central nervous system of the C. elegans, which part is actually responsible for the generation of some movement, both crawling and both types of the locomotion. But here again, they discovered maybe showed that the L1 is lacking some type of interneurons which are for the adult one is responsible for generating forward and backward patterns. Here in the L1 only it is actually using 3 of those 6 nodes and interneurons for generating the pattern. So we can maybe conclude that somehow not having enough maybe interneurons during the L1 could be responsible for that type of not sustaining movement patterns. Also, we have another paper over here. If other one are interested, this is the main paper that the OpenWorm project, especially its muscle cell, is trying. Actually, the body wall muscles and our simulation that we have done and the code available is based on this work. Somehow we can see all types of the formulation of those patterns that we have we could see that are responsible for generating the movement patterns. It is somehow the electrical the equivalent circuits for those patterns, and we have used this, and we have the code available on OpenWorm GitHub. And as I described, somehow I'm going to use some similar maybe approach to try to understand maybe the difference between the L1 and the larval stage and the adult stage of the C. elegans, difference in movements, and specifically trying to and understand how different neural networks in the C. elegans during its larval stage and its adult stage is going to make some distinguishable pattern generations, and so somehow some artificial neural networks that could be derived from such a work.
Great. That's great. So distinguishable means like, a specific function, or just like kind of distinguishing it with the eigenworms, like that they're different.
Of course, if we are going to quantize the and as you saw and you described here, the quantifying the locomotion is of course related to the different eigenworms, and if we are going to work on that, maybe the number of different eigenworms that are somehow related to maybe different types of neural networks, and combination of those neural networks maybe could somehow move our work from a simple model, which is a worm in its L1 adult, it's in its L1 larval stage actually tries to generate the patterns, brings us to a more complex one, which is of course the adult one.
Yeah, I think there's well, what I was thinking was that there are data sets where like they look at synapse development in the larval stages. So there are data sets that exist that show kind of like the idea in larval stage worms is that there are these synapses that sometimes they're persistent, meaning that you get an early synaptic connection, and it persists throughout development and into adulthood. Some of them, however, are sort of transient, meaning that they kind of are there and then they go away. And, so there's this data set that I'll have to kind of go over in more detail later, where they have these characterized for different connections between different neurons. So we know the connectivity of the connectome from, different, basically from gap junction studies. So they look at like the cells, if they make contact and they have a gap junction, that's a connection. They do this from microscopy work. So they look at the cells, they track the cells and where they have junctions. The other way you can do it is look at the synapses, and that's harder, but it gives you a different map of connectivity because it tells you kind of what maybe functionally is going on. So these changes might very well be reflected in some of the synaptic wiring that happens and changes.
Yeah. And actually, another approach would be maybe molecular level, because we are both working on cellular level and on maybe the construction of of all these compartments. At the end, I'm trying to somehow compare everything that is going to be related to cellular level, especially ion channels, and those which are at the network level, which are usually the wiring of those interactions between muscle cells and neurons. So I'm trying to have both points of view.
Have they looked at muscle twitches in relation to the development of the connections in the brain cells?
Well, the muscle twitches that you see in the embryo, it's kind of hard to get a handle on that. I mean, you get like when the connectome starts to wire up, as it were, you get like a few things wired up, and then you get a lot of things wired up, like in a very short period of time. So like the muscle twitching is kind of hard to know kind of what's happening. It's probably just kind of as the muscle cells differentiate, and they're syncytial cells, so they have multiple nuclei, but you get these muscle cells that differentiate, and then they get contacted by neurons, and then they maintain the muscle so it doesn't like go away. And then all of that, there's twitching that happens so the muscle can sort of get practice firing, firing and being active. And then, when the connections are made with the connectome, it starts to get refined. But yeah, I don't know what's happening like in L1 versus, I don't know of any data set in like the embryo that tells us anything about muscle twitching. Maybe like there's some gene expression that says, oh yeah, there gene expression data that says, oh yeah, late embryo, maybe there's some muscle genes, muscle related genes that are really highly upregulated versus like L1. And actually, I know that those data sets exist for those things. So those are kind of interesting. I might take a look at that. I have the data. I've been working with the data sets for another paper. That's why I bring it up. But
Oh, okay.
yeah,
Great.
yeah,
Yeah, muscles and nerves kind of go together, like they're like part of the same system.
yeah. Well, that's great, Vahid. So they don't have data sets for what they're doing. Yeah.
Yeah.
So but what would you say is like the next thing you'd like to do there? Like, I know you've been kind of working on this big review document.
If you could have those, I'm still looking for any new data or something that could be digitized even from some old papers. If there could be any data available, I may try to somehow use the same approach we use for the adults. Actually, simulation of the locomotion. Try to use some similar one for the L1 stage. However, here we need some maybe some data that could put it in a way that both the neural networks of the system and also the mechanical model, the biomechanical model of the system could be described very well. We are trying to understand the equivalent electrical circuit of this simulation, something similar to the paper Boyle and Kwan I presented, but we are trying to minimize that in the level of L1 stage. So if, for example, a recorded L1 stage is somehow has some movements and some locomotion slower or something different with different patterns, we could understand how different maybe combination of neural networks that are responsible for the locomotion could be again, I'm saying we minimize the less compartment we are going to use in a new maybe model could be described how maybe a minimized model of a locomotion could be enough for the locomotion of a C. elegans, and maybe some other networks are responsible for different behaviors of the C. elegans. So this is something that is the main point of interest in such a study. I'm still looking for data as well, but trying to see if it is possible to estimate some of them as well, especially regarding the size of the C. elegans in the larval stage and the L1 stage. So we could maybe estimate that with that much, that size, for example, if the L1 size is less than, I guess, half of the adult one, and the number of muscle cells which are less than the both muscle and neuron cells which are less than the adult one during which are responsible for the locomotion. So how it could be estimated that with that much data and required compartments, we could regenerate the same model. So it would be both the validity of the adult muscle model that we have, and again, as I'm saying, the minimal requirements for generating something like that.
Yeah. When you say minimal requirements, you mean like for the worm, or?
Yeah, for sure.
Yeah, like it would be like what the worm has to do minimally to.
Yeah.
So like, yeah, we don't know like what it's actually doing, but we know what it has to do, basically.
Yeah, there's lots of simulations that we are dealing with right now. Is somehow maybe lots of them are estimated. So one more thing we need is to, if we are going to understand or to model the C. elegans during different larval stage, we would need. It's something like there is a sample. There's a actually. It has been mentioned in one of the papers that I've been describing that it somehow looks like the learning mechanism in our children. We can say a child during different stages how it could learn to use different neural networks for tracking, for running, and any behavior that they have. We could use some similar points of view for the C. elegans. Different larval stage means that different learning algorithms are working in the C. elegans to so that it could learn how could better swim or better crawl, and this is the main point for this study, of course. So adding different parts or components at each stage could make different models more robust, and at the end it could say that at each larval stage what new maybe learning patterns are going to be generated.
Yeah, that's great. So does anyone else have any thoughts about that? I don't know if Morgan had any thoughts on it, or if anyone else were curious about, like, had questions for Vahid.
Do the worms learn? They learn, do they? They have a memory?
Yeah, they have a memory. I know that people have done associative memory studies in worms with food and things like that, but I don't know what I don't know like too much about the learning studies other than that they exist. But yeah, they do. They can learn, and of course they have like acquisition and everything. I don't know. People have done like in larval development versus adult though. Again, interesting. All right, so that's great. Thank you, Vahid. I look forward to seeing more on that. I've been looking over your document. I don't have anything to add right now, but I'll be working on it. It might take a while to get through it. Yeah. So yeah, Hussain had to leave. Thank you, Hussain, for attending. There's a set of papers that are coming out, and so it's on someone called Erwin Bauer. So Erwin Bauer was a very early theoretical biologist. I think he was like one of the first theoretical biologists, and he was from Eastern Europe, from the Soviet Union and Hungary. And so he did a lot of things with theoretical biology, and there have been a number of papers that are coming out sort of in honor of Erwin Bauer. And he has an interesting connection to a lot of the early complexity theorists as well. So this is his Wikipedia stub, and talking about his research here. So he was one of the first theoretical biologists. He was interested in the general thermodynamic features of living systems. So he published a monograph in 1935 called Theoretical Biology, where he goes over this stuff. And so he...
Bradley, are you sharing your screen?
Oh, yeah, let me share my screen.
Oh, thank you.
Yeah. All right, there we go. So this is his Wikipedia profile. And so he formulated the principle of stable non-equilibrium state, which is like a concept that's very similar to like non-equilibrium thermodynamics and other concepts that people have come up with, like dissipative structures and things like that, if you've heard of those terms. So there are a bunch of people at this time who are kind of working on these basic ideas, and so they're kind of percolating at this point. So he formulated this principle, which he considered as the basic characteristics of living matter. According to Bauer, living systems function at the expense of non-equilibrium, and the external energy is used not directly to perform work, but to support the stable non-equilibrium state. Bauer's principle is incorporated into nonlinear thermodynamics, or the nonlinear thermodynamics of irreversible processes. So irreversible processes just means that, when we talk about something like entropy and the second law of thermodynamics, that those processes aren't reversible. You can't go backwards in time. You can't go backwards in the process of entropy. So he had a lot to say about that, mainly with respect to like open versus closed systems. So a lot of stuff having to do with the second law of thermodynamics involves closed systems and characterizing closed systems. And what happens, in life, of course, is that it's a very open system, meaning it doesn't have, that you always have energy coming into it. And so most, characterizations of the second law involve sort of no energy coming in. You have a fixed amount of energy and you watch things run down to entropy. And that's the way you characterize it. So this is how we've characterized entropy traditionally. What he's saying is that living systems are very open, there's all sorts of energy coming from different places. And so you have a lot of things that we have to sort of, biology is distinct from physics in that respect. And so that's what he's getting at with this. So living systems in this framework cannot support their organization only due to the influx of external energy, an example being the ordering internal factors involved. So the activity of living systems is fully determined by the internal pattern of its non-equilibrium state, and any work performed by the biological system itself appears as the work of its structural forces. So evolution, according to Bauer, corresponds to an increase in external work, which aims to exploit additional resources to maintain evolving biosystems. So again, he's like distinguishing this from physics, where you have closed systems. So he was actually a contemporary of various complexity theorists. And so there's a paper here. This is the DOI here. I can't remember where I got this. This is a screenshot of it, but I couldn't get access to this article. This is The Organism in the Open System, Erwin Bauer and Ludwig von Bertalanffy. Bertalanffy was a complexity theorist and an early theoretical biologist as well. And so they talk about the different connections between these two people's work. Howard Petit was also an early theoretical biologist. And so all these people had very compatible ideas, and they were kind of hitting upon the same thing, but wasn't quite all sketched out. In fact, it's not all sketched out now, which is what I'll get to with the actual paper that I'll be talking about soon. So the abstract here is that in this historical treatise, two biological system theories formulated in the 1920s and 30s are roughly sketched. The first part discusses the concept of a thermodynamically open system. It is coined by the Russian pathologist Erwin Bauer. Like Bertalanffy, Bauer wanted to prove the specificity of the biological sciences against physics. So they wanted to show that biological sciences were a specific instance of physics. They were separate from physics, and maybe they have their own rules that exist. So, it's not that biology is non-physical, it's just that it's an extension of what we know from physics. To achieve this, he postulated the necessity to formulate specific laws of motion, which are valid for living matter alone. So this is where we go from like the second law of thermodynamics to like, say, the first law, which is a law of conservation. Or, there are other things that the laws of motion are constrained in physics. Trajectories are constrained by the energy consumption and their entropy. And of course, what he's saying is that living systems have a different, because they're open systems, they behave a little bit differently. And this is important, of course, in reproduction and in some of the energy consumption patterns of living systems. So we know that living systems, do a lot of things at very minimal energy. So this might be one of these things that they're trying to get at here.
I have a whole book on hypoxia.
Oh yeah.
Yeah. Cells behave differently depending on the level of oxygen.
Okay.
And
yeah.
the activity of the mitochondria, like that's connected. And like they're stiffer without oxygen. It dramatically changes the mechanical properties.
Yeah.
So I've been trying to get at that with the things I'm writing, because if it's alive, it behaves differently than if it's dead. So if you take a piece of tissue out of a living creature and measure its mechanical properties, you are not getting the mechanical properties of the live and living creature that you're measuring. Yeah. So there's this whole huge book on hypoxia.
Yeah. Yeah. So and then in the second part of the paper, the organismic system theory of the Austrian-Canadian philosopher Ludwig von Bertalanffy, Bertalanffy, is outlined. The focus of this theory relied on the process dynamics that is inherent inside an organismic system. So they're talking about like an organism and the process inside an organism, and that it's an open system, so we can't model it as like a closed system where an organism serves as the boundaries of an organism serves as the boundaries of the system. And so they kind of get at some of these processes within the organism. And remember, this is back in the mid 20th century or even the early 20th century. So this is not, the latest kind of data that we have on molecular biology or metabolic networks or any of that. This is just kind of like, what we think is going on inside the organism. Both theories exemplify closure models for a living organism from a methodological point of view that distinguishes these earlier models from something called semantic closure, developed by Howard Petit as an epistemic clue in solving the enigma of living phenomena. So they get into this. This is kind of jargony, but they get into some of the ways that you might model this and how you might consider the organism as having these biological boundaries as being sort of semi-closed versus like the influxes of energy which keep it open, and having these sort of energy these production requirements with respect to the energy consumed in an organism. So the objective here is to disclose the essential differences between the conceptions of closure, and so this is pretty technical stuff. To encourage further research on closure, the essay concludes with a few questions concerning clarification of the terms. So this kind of goes through a number of citations here from older works that are relevant here. So people have been working on this sort of physio-mathematical foundation of biology for quite a while. It's just that we have all these concepts out in the literature, and the question is, how do we pull them in and like kind of make use of them with the modern data sets that we have. So, there are all these tools, and so that's what we're doing here. So this brings us to the modern day, and so there are a number of papers coming out. Our friend here, Abir Igamberdiev, who we actually, Dick and I published with him in a special issue a couple years ago. He put out this paper called Towards the Relational Formulation of Biological Thermodynamics. So this is about, this is using a specific sort of approach to looking at biological thermodynamics, is this unique thing. So there are a couple other papers in memoriam of Erwin Bauer, so I'm not going to get into those papers, but I'll talk about this paper a little bit and then finish up the meeting. So the abstract reads, classical thermodynamics employs the state of thermodynamic equilibrium, characterized by maximal disorder of the constituent particles as the reference frame from which the second law is formulated and the definition of entropy is derived. So this is what he's talking about, is like the classical statistical mechanics model of where you have like a landscape of particles that are, at different energetic states. And you start out there and you have a closed system. And then the energy decreases as time goes on because there's no new energy coming in. And those particles, being in all these different energetic states, settle into minimal energetic states. And those minimal energetic states define a landscape that defines this sort of entropy profile. So entropy is where things kind of go from these different energetic states to disorder. So where they're in these minimal energetic states, the implication being that there's structural order in the original state, and then you run down to this minimal state. So this is the classic thermodynamics view of entropy. And this is not something that you see in open systems as much because you have constant flux of energy in, so that energy landscape gets disrupted and you can maintain some of that structure. So non-equilibrium thermodynamics analyzes the fluxes of matter and energy that are generated in the course of the general tendency to achieve equilibrium. So the whole point of life, I guess, is to achieve equilibrium. So when you have energy coming in, it's a means to maintain equilibrium. So systems, like for example physiological systems, are often said to be in homeostasis. And homeostasis is this equilibrium with respect to thermodynamics. Now you have non-equilibrium thermodynamics because you have fluxes of energy. So these fluxes of energy kind of push, besides providing energy for the living system to keep it from running down to total disorder, you also have these fluxes of energy. And the consequence of that, and because we have fluxes of energy from the environment, that's how it comes packaged in like sunlight or in food or whatever. And the challenge for the organism is to maintain a stable state in the face of all of that. So that's what he's getting at there. The systems described by classical and non-equilibrium thermodynamics may be heuristically useful within certain limits, but they have there's a fundamental problem in the application to living systems, and he mentions autopoietic living systems, which is just a way to say that the complex system is trying to maintain some sort of equilibrium. We discuss here the paradigm defined as a relational biological thermodynamics. The standard to which this refers relates to the biological function operating within the context of a particular environment and not to the abstract state of thermodynamic equilibrium. So this is just kind of thinking about the environment as the thing that sets this biological function of homeostasis. This is defined as the stable non-equilibrium state following Erwin Bauer. So this is kind of bringing back to Erwin Bauer and his stable non-equilibrium state. So that's what he calls this, and it's basically like homeostasis or something similar. And like I said, these terms were all being used by different people back in the early to mid 20th century. So there's some discontinuity in terms of, a formal theoretical model, but they're all basically getting at similar things. So similar to physics, where abandoning the absolute space-time resulted in the application of non-Euclidean geometry. So when Einstein came up with his general relativity and his relativity concepts, they abandoned this idea of the ether or of absolute space-time with respect to how the universe was structured. So this is a relation. He thinks of this as a sort of a relational way of doing this, and then he says, okay, we can do the same thing in biological thermodynamics and have this sort of relational biological thermodynamics. And this may lead to revealing the basic iterative structures that are formed as a consequence of the search for an optimal coordinate system by living organisms. So these living organisms are trying to put everything into this framework of, maintaining a stable non-equilibrium. And so it's relative to the organism's environment, to the organism's structure, and so forth. Through this search, the developing system achieves the condition of maximization of its power by synergistic effects, which is a fancy way of saying that there are these, non-linear effects that happen when things interact, and that gives it the power to do this sort of maintaining its sort of non-equilibrium thermodynamics. So this is nice. This is a nice tour of some concepts, talking about Ilya Prigogine and some of his thoughts about the second law. And, this is, again, another person who thought in terms of biology being an open thermodynamic system and kind of the consequences of that. And so this is a nice paper. It ties together a lot of concepts. And I'm not going to get into the details of the paper, but to say that this is something people have been thinking about for a long time. And, maybe there are some updates to this. And it looks like people are publishing updates on it. And it's interesting to see what they have to say. So I see that Susan posted a link to some hypoxia work that's been done, some methodological work. So hypoxia is a good example of like energetics and maintaining homeostatic state with respect to energetics and oxygen consumption. A lot of interesting work there. We think we know about things, different physiological functions, but a lot of times we know very little because we have sort of idealized data in a lot of cases. And so, sometimes you have individual variation in how people metabolize things. Sometimes you have people who perform at a very high level relative to the rest of the population. And then, of course, we have different species where they have different specializations for dealing with low oxygen environments, like in deep sea organisms and things like that. So they're different ways of doing this. So then Susan asked, what system does not go to disorder? Answer, life. Yeah, so life doesn't go to disorder until it dies, and then it's disordered. But it has to maintain life, and so that's a good question about life. You say, what is life? It's non-equilibrium thermodynamics, but what does that mean? It means you're not dead, I guess. So, and then, it's like a non-answer. It's like, I'll define it when it's not dead. It's like, so that's where we are with that.
I gave a physics teacher a bad time with that one.
Yeah, so they're physicists who have a very kind of constrained view of thermodynamics, and then you have biophysicists who have to deal with applying that to an open system, and then you have people like philosophers in that who talk about thermodynamics of life, but then don't actually deal with like experimental data. So it's like we have these different perspectives on it, and getting it unified is, it's tough.
Part of this is the change of pH changes the way proteins are folded, and so you change the oxygen level, and it changes the pH, and that changes the molecular level of things. It kind of fits together. I have another paper. It's more like a thesis that somebody wrote about intermediate filaments, I think it was.
Okay, yeah.
And she was talking about pH as being a factor in that stress strain curve.
Right.
You can get.
Yeah.
It tell you the elasticity of various molecules. Vermentin, I think she was into looking at vermentin as one of the main intermediate filaments. Do you want that reference? Like,
yeah, that would be good. Yeah.
I'll try to look it up.
All right. Yeah. I know we've talked about intermediate filaments before, and it's interesting. So in that case, we have like an environmental condition that actually sets sort of how that stress strain curve looks. So it's like all the things about the biophysical properties or the mechanical properties of whatever you have is, mediated by this factor in the environment. So different organisms, perhaps with different pHs, they behave differently, or in different chemical regimes.
Yeah, I think that one of the more interesting things is the difference between warm-blooded and cold-blooded animals.
Yeah.
So yeah, that's also interesting. Let's see. I just have to look this up. It starts with S, and I have to find my references.
Yeah. So while Susan's doing that, does anyone else have anything?
Sheffers. Anyway, I'll write it down.
All right. Does anyone else have anything else to say while she's doing that?
Just the usual origin of life kind of perspective on this in terms of the generation of it's not just the generation of biomolecules, or it's the protocells and metabolism as being the two kind of key components, and their work to maintain the ordered system.
Yeah. That's good. Thank you. Yeah, I didn't bring up early life, but we've talked about that in the group, and one of the reasons we are interested in that is because it's, well, first of all, it's a simple system, kind of like C. elegans, but also because, it does give insights into sort of this biological regulation, or these regimes of regulation, which maintain a biological system. So that's something we don't really do much in OpenWorm per se, is kind of get at that question. Like, we don't have like a physiological model of sort of how C. elegans maintains its state, but it's it's part of life, so it's part of simulating life. Would be simulating those conditions. So. All right. I guess Susan is coming up with some answers in the chat here. There we go. Any other comments before we go? It's like Anna Shepard's 2022 intermediate filament mechanics across scale from single filaments to single interactions in networks and cells. So yeah, this is a sort of a specialized publication, but it's something, if you're interested, you can look it up. Okay.
Yeah, I thought it was interesting.
Yeah.
It covers more than fermentin, but that was the main filament I noticed. I think there was also keratin in there.
Okay.
Yeah.
I haven't mentioned Bradley. I never did a good synopsis of the advanced microscopy workshop at Berkeley.
Okay.
Before Photonics West, I was just thinking about some of the imaging that they did of filaments. Susan made me think of that, and just some of the other kind of techniques they had for looking at kind of more specific aspects of the cell infrastructure and things like that. That was focused exactly on imaging microtubules, cell. What did she call it? I mean, it was something like cell infrastructure or something like that, but it was really like the imaging of filaments, microtubules in action, and lots of kind of biophysics. Biophysics of the cell. I can totally get that. Yeah, I'll find that. I'm actually still on the school run, kids school run here. So
That's okay. Yeah.
soon as I've got my hands available, I'll check that. But but lots of great talks, and in particular, I just wanted to say the head of the Chan Zuckerberg BioHub imaging group, Loc something, LOIC. Oh, I'm blanking on his last name.
Royer.
Anyway, he had a great demo of Chat GPT and Pari. You know, that Python framework for microscopy that that Chan Zuckerberg BioHub kind of I don't want to say I forget who exactly wrote it, but anyway, but they've been a big proponent of it, a big funder of it. Anyway, it's just it's like an imaging. It's kind of like a Fiji, ImageJ like app.
Yeah.
And he was just showing like ChatGPT writing code for plugins in Impari, and it being both a great way to learn how to code as well as to generate functionality. It was really cool. Anyway, there was lots to cover in that would be good to go over.
Yeah. Well, I have one image of a cell, and it shows a microtubule mat all the way across the top of the cell. And then I have others where there's just actin there, and I know they both occur, and sometimes more one than the other. And I would be very interested in having more pictures, more images. That's what I'm actually missing. Is I'm trying to make a structure, and I don't know what it looks like in real life because there's no 3D images of the inside of a cell.
Sure. Now, this was like the focus of her lab's work. Yeah. So I'll find her name. I mean, pretty sure. I want to say she's a Berkeley prof. But again, I'll get to that. No worries.
Okay. Yeah. Kind of important here if I had some more 3D images of cells, actual cells. It would be great.
Yeah, we should go through, the ImageJ's Fiji ecosystem at some point, because there's a lot of stuff where there's open source tools everywhere, and it's just kind of hard to.
Yeah. No, I only kind of discovered some frameworks recently that are. I don't know if I want to say built on Fiji or kind of built around it in the sense of, almost like an app store for Fiji.
Oh, okay.
Yeah.
Yeah, I've used ImageJ before.
Yeah, so I mean, I remember ImageJ as an undergrad, but it's looking at like what Janelia and again most of the people who were covering the image processing pipelines at this workshop were focused on Fiji.
Yeah. Interesting.
Yeah. And
for the work for some data I have, and I put it into Fuji, and it overwhelms my computer.
Well, so this check out. I want to say it's NPARI, like NPARI.
Yeah.
I forget if it's like funded by BioHub or it was originally them, but then funded by because it's Python tool, it might be focus. It might be NumFocus funded. I forget now, but
okay. Yeah. I think it may be. Yeah. Yeah, that's great. Yeah, so thanks. Yeah, that was great. I'm glad that you had fun at the workshop there, and it sounds like it was. Maybe we can do more formal thing. I mean, even our Saturday meeting, if you want to do that.
So let me know if you're where it's happening.
Okay, yeah. All right. Well, thanks for attending, everyone, and see you next week.
Okay, bye.
Bye.
Thank you and goodbye, everyone.
Thank you. Bye.
Yes,
Thanks.
bye. Yes, there. Bye.
Bye. So now I'd like to follow up on a paper I talked about last week. And so last week I talked about this paper, three-dimensional soft streaming. And this was mainly a materials science paper that talked about fluid mechanics and fluid dynamics with respect to something called viscous streaming. So this is something where there's a sort of a streaming of liquid around a body, and they talked about soft-bodied objects, and they talked about some things that were related to biology. And so it wasn't specifically a biology paper, but it had a lot of things to do with the compliance of soft materials and the kinds of things we would see in tissue morphogenesis and in tissue mechanics. A lot of that was about flow control. And so that sets up this paper, which is much more sort of oriented towards biology. So this is a paper out of Nature Physics. This was published online in January, so last month. And this is called Self-Organized Intracellular Twisters. So here are the authors here. And I'll go through the abstract and we'll kind of see how these two papers form a theme. They're talking in this case about different types of flows that are connected to this kind of streaming activity. They're going to talk about this in the Drosophila oocyte. So the abstract reads, life in complex systems, such as cities and organisms, comes to a standstill when global coordination of mass, energy, and information flow is disrupted. Global coordination is no less important than single cells, especially in large oocytes and newly formed embryos, which commonly use fast fluid flows for dynamic reorganization of their cytoplasm. These cytoplasmic streaming flows have been proposed to spontaneously arise from hydrodynamic interactions among cortically anchored microtubules loaded with cargo-carrying molecular motors. So they talk about cytoplasmic streaming flows. And so these are streaming in the cytoplasm. These are constantly moving flows that are occurring. They spontaneously arise from hydrodynamic interactions among microtubules that are anchored, loaded with cargo-carrying molecular motors. So the molecular motors are using this cytoplasmic streaming flow. They arise from these hydrodynamic interactions within the cell. And that's where we're going with this. So we had the idea of streaming in the last paper. This paper we're talking about the same kind of streaming idea, but in the cytoplasm specifically. Here we combine modeling and simulation with live imaging to investigate such flows in the Drosophila oocyte. So this is in a model organism, Drosophila oocyte in the embryo. Using a fast, accurate, and scalable numeric approach to investigating fluid, structure interactions with thousands of flexible fibers, we demonstrate the robust emergence and evolution of cell-spanning vortices, or twisters, in three-dimensional cellular geometries. So now they're talking about, how these things emerge and maintain themselves, and they are these cell-spanning vortices or twisters. So there are these vortexes that exist within the cell, and they go across the cell, and they sort of coordinate these streaming events. And so they actually model the geometry of this in cells, which is important for really being specific about how these streams affect morphogenesis. These twister flows, dominated by a near rigid body rotation with secondary toroidal components, reproduce the variety of experimental observations. In cells, these flows are probably involved in rapid mixing and transport of oplasmic components. So this is something that is involved. It sort of allows for transport within the cytoplasm, but also mixing of things within the cytoplasm. So you can, the molecular motors have to move to different places in the cell to do their work, and of course they need some sort of highway to do that. So the streaming mechanisms within the oocyte allow for that. You also have mixing, so you sometimes need to have things well mixed. You don't want things segregated out as part of the, different parts of the cell, so you want to do that as well. So this is an old concept, cytoplasmic streaming. This was first described in 1774 by Bonaventura Corti, who was, I guess, an early microscopist of the cell. Regulates a wide range of intracellular processes, especially when large cell size makes diffusion and motor-driven transport too slow for efficient intracellular transport and mixing. So if you have a cell, you have things going on inside the cell. And, they're throughout the cytoplasm. Things are moving from the nucleus to the membrane, and from the membrane to the nucleus and to other organelles within the cell. So you have all these transport imperatives. And so streaming allows you to coordinate these to make sure that things aren't compartmentalized or they don't cluster in certain locations. Now, when they talk about diffusion, of course, we've talked about that with respect to the cell. One common explanation is diffusion or Brownian motion. And diffusion, at least Brownian diffusion, operates sort of locally where some particle or some thing, it could be a molecular motor, explores the space around a common central point. So the path I'm drawing here is sort of the path around that point. And as you can see, it doesn't really do a very good job of transporting things throughout the cell, especially if the cell is large, if you have a lot of things you need to coordinate in a directed manner. So Brownian motion is not a good explanation. But if you had flows or dominant flows that kind of move things around like this, mixing them, or things moving outward and inward from the nucleus, that might be a better way to transport things. You can combine that then with Brownian motion and get this sort of system where you get this flow, this dominant flow mode, this streaming, but then some variation along that stream. So the stream is kind of like, has a little bit of a wiggle to it, and it allows for changes to occur. So this is about transport, but more importantly, it's about efficient transport, something that doesn't take forever to eventually diffuse out to the edge of the cell. And with Brownian motion, you not only get this just kind of blind exploration around a single point, it sort of migrates, not necessarily directionally, but it migrates directionally to some extent, but it's very slow. And so that rate is too slow for a lot of the metabolic processes in the cell. Then the question is, how does this mechanism occur? We know it's useful. We know that it can allow for circulating flow and moving things efficiently and quickly to certain destinations. And it doesn't rely on this slow diffusion process. But it has to be set up by some mechanism. And so cytoplasmic flows are commonly driven by forces originating from the cell cortex, where motor proteins carry cargo along cytoskeletal filaments and so entrain the fluid. So, of course, your cytoplasm is fluidic, and you have something like this, where the cytoplasm is in between these two lines, and you have filaments that sort of coordinate. So, the filaments sort of order or organize these flows to go between them or through a set of filaments. And so, you might have something like this, you might have something like this, where it coordinates the movement. The formation of macroscopic flows in cells requires an alignment in motor movements across many filaments. In some cells, this alignment originates from the organization of actin filaments at earlier developmental stages. So actin filaments are organizing themselves in different ways. In this case, they're moving things with their, they have this, actin filaments expand and contract. And so they act like little muscles moving things. This model of algae, they have this sort of developmental origin, and it can provide a static boundary condition for the flow problem. In others, like oocytes of the fruit fly Drosophila, which is the one they're going to focus on here, Motors move on a cytoskeletal bed of flexible microtubules, and their alignment and direction were proposed to arise by means of self-amplifying feedback between motor-induced cytoplasmic flows and collective deformations of the microtubule bed. So in this case, we have these flows that are liquid and they're moving at a certain rate, but then we also have these deformations of the microtubule bed. So this is my diagram of the filaments organizing themselves. The microtubule bed might be something like this, which can be deformed, so it can have some displacement, and then you have the flows themselves which move against it. And so all these things interact to provide a mechanism for movement and a mechanism for facilitating movement. So the cytoplasm of the developing Drosophila oocyte remains relatively quiescent for the first 3 days of oogenesis. So this is like very early development. During this time, diffusion and directed transport. So this is a combination of diffusion and directed transport. So you have diffusion on one of these flows like we showed here, and those things combine in a way that allows molecular factors to localize and get to the place that they need to go for patterning. So what we have basically is a model where flows can disrupt sort of this compartmentalization, but it can also promote compartmentalization and patterning. So this is a better sort of visualization of this than I was drawing. You have these nice animations here. This is a stable substrate, this is a beating substrate, and this is a streaming substrate. So you can see that these filaments sort of are organized in different ways. So when it's stable, they're all sort of aligned. Beating is where they have this pattern. It's kind of like one of those air dolls that they have where they push air up into it and it dances around with the pulsating of the air. So it's this beating. It's not very coordinated, but it can, move around. It can move things. But that's still kind of, sort of akin to Brownian motion. It's not really great for moving things directionally. In the case of streaming, these filaments bend in a certain direction. So over time, there's bending in one direction instead of like bending in various directions over time. And then, of course, in the stable case, you get no bending. It's laminar. And so you get this, streaming is of course, the better option here. So this shows where you have beating, streaming, in a stable regime. So you have this phase diagram that show these different cases. So beating is this part of the phase diagram, stable is here, and streaming is up. Here's another example here of what these look like on the membrane of the cell. So you have these actin filaments that are bound to the cortex, and so you can see that they form this sort of layer of filaments that can facilitate streaming or impacted by streaming and sort of facilitate the flows. And so in this case here, so this is case 1 and case 2. Now if we go back up to the phase diagram, case 1 is in beating. So this is where they're beating and they're moving around as you saw up here. In case 2, they're streaming, so they're all coordinated and oriented in the same direction. So you can see that they might facilitate a flow that's operating with respect to streaming. So streaming is not just the fluid flow, it's the orientation of the substrate as well. This shows an example here of case 1 and case 2 in time. So this is a diagram that shows the flow, and it shows the physics of the flow. And so you get this much more coordinated flow that goes around sort of in a circular manner. So this actually shows you, some of these properties, some of the dynamics going on in the cell. And then, of course, we can see examples of this this beating versus streaming. So this is beating, this is streaming, and of course, the physics of it are much more ordered here than the dynamics are much more ordered in the case of streaming than beating. Beating provides these oscillations, streaming provides this more linear state. They need to model these things, so we have some nice animations here, but they're going to do some computational modeling of hydrodynamically coupled motor-driven fibers. So conceptually, plus N directed motors, here they use the kinesin 1 molecule, bind along anchored microtubules. So they're binding kinesin 1 to these microtubules that are anchored to the cortex. So they call them plus N directed motors, meaning that there's an end that allows for the motor to direct its cargo. So these plus end directed motors, which is a kinesin 1 and an anchored microtubule combined together, they carry a cargo towards free plus ends and detaching once they reach there. So they move things from one place to another using the ends of the microtubules. Although the cargoes seem varied, possibly inducing cellular organelles and yolk granules, free microtubules have been identified as one cargo crucial for robust streaming. So this kinesin 1 is a motor protein, and it moves along these microtubules. And of course, we need to be able to carry cargo, and to facilitate the streaming, we have to have the right sort of molecules in place, and the right molecules doing the driving of this. Given the lack of data on the details of cargo binding and payload densities and sizes, we know the mechanisms, or they're proposing the mechanisms, and we can use that in the modeling, but we don't necessarily know a lot about how different payloads operate, how efficient this mechanism is, and so forth. So if you think about a highway with trucks on it carrying cargo, you might know that the trucks ride on the highway and that they can deliver things from place to place, but you don't necessarily know how efficient it is for every kind of load that you might put in those trucks. So they can't necessarily use that for modeling, but they can just assume a simple model. And they cite reference 24 here, where they coarse grain the forces of plus end directed cargoes on bound microtubules to a uniform compressive force density. So that is directed along the bound microtubule towards its anchored end. An equal and opposite force is exerted on the surrounding fluid, thus satisfying Newton's third law. We find this is sufficient to recover many aspects of observed streaming and to make several predictions. So consider end microtubules clamped to the inner surface of a spheroidal cell of effective radius r and surface area s. And so this is where we have, again, the inside of our cell. And so we have our cell here. We have the cortex here with the microtubules kind of lining this. And so now, according to the paper, we consider end microtubules and clamped to the inner surface of a spheroidal cell of effective radius r, surface area s. So the surface area is this area here, and then r is the radius out to here. So we need all these parameters to model this. So it's a cell of a certain size, of a certain radius. There's a surface area, so cells aren't usually just sort of spherical. They have, different shapes and different variation there. And then you have a number of microtubules, which you can count as n. So the larger the surface area, the more microtubules there are. The larger the radius, the more microtubules there are, and so forth. This isn't necessarily going to scale linearly, but we can get a sense of what it should look like using these three parameters. Microtubules are well modeled as inextensible, elastic, slender bodies, and so this is where we saw these little tubes up in Figure 1, where they were either like kind of twisting around in the wind, as it were, in the flow, or they were bending in a coordinated manner. But they have this sort of flexibility, this elasticity that allows them to bend and move around. But they're anchored at one end, remember, so they can't just move freely anywhere. And the cytoplasm is modeled as a Newtonian fluid of viscosity mu, which is from reference 22. The shape of a microtubule I at time t is given by xist, where the s is the arc length from the base and li from its length. So basically they're modeling the shape of the microtubule. So this is where we have, here we have the microtubule, which is anchored at one end. And it's bending, and it has physical properties. And then the force is here, the flow is here, and there's a force. So this is a Newtonian fluid, which means it should respond linearly. It's not a non-Newtonian fluid, which is nonlinear in its response. And then this is the filament here, which is use several parameters to characterize the filament. The shape is given by XIST. The microtubule shape evolution caused by drag forces balancing elastic and motor forces given by its local slender body theory, which is this term here. And so we can model this as a physics problem as this sort of this thing that's kind of facilitating the flow. It's bending with respect to the flow, and there's a transport that's going to happen in the system. Simulating the system efficiently for thousands of microtubules has peculiar challenges. The microtubules make the system geometry very complex because you have this, the surface isn't constant. It has, bends in it, and it has, variations in the surface. And so the microtubules line that surface and they bend. Sometimes they don't always bend in the same direction as we talked about. So sometimes they're not always in line with the streaming. But they all have, we have these independent filaments, and maybe they're not uniform down the surface of the cortex. So we have these, multiple filaments, and sometimes they're aligned and sometimes they're not. But this is the challenge in modeling this. Sometimes the cortex is undulated, sometimes it's a straight line. So although the number of degrees of freedom, mainly discretized microtubule forces and shapes, is not extreme, so it's around to the order of 10 to the 5th or 10 to the 6th, which is manageable in a computational environment, modern computational environment. All are globally coupled by the Stokes equations, and the system needs to be simulated for long times. So we have the Stokes equations, which model the fluid dynamics, and so we need to have this. For this, we developed a fast and scalable computational platform that accurately evolves equations. This is from citations 1 and 2. It has 3 main components. So the first one is that the boundary integral representations in slender body theory reduces the 3D Stokes equations in this complex domain to solving one-dimensional integral-differential equations on microtubules and a coupled 2D integral equation on cell surfaces of nearly arbitrary geometry. So they simulate a number of geometry, cell surface geometries. They model, they couple that with the microtubule model, and they get their boundary conditions. Second, the fast Stokes solver efficiently evaluates the non-local hydrodynamic interactions between microtubules and the periphery with linear scaling in the number of unknowns. So again, you have these different models modeling different components of this relationship across different cell geometries. So we can do this with a fast Stokes solver. And then third, we use a stable implicit explicit time stepping scheme to efficiently evolve the stiff microtubule dynamics. So the microtubules aren't just of a constant stiffness, they can have a differential stiffness. This allows us to have this streaming mode or other types of states as well. And so they characterize the streaming phase and they characterize the twisters. So the twisters are global phenomena or consequences of the streaming phase. So henceforth, we focus on the streaming phase, which generates flows similar to those typically observed in oogenesis, stage 10b and beyond. In this region, we find that the initially straight microtubules at first bend in seemingly random directions. Then gradually they align into an array of bent microtubules wrapping around an axis of symmetry, which you can see here in G. So you can see this, how this model begins. You have this bending in different directions like this, and then you have this coordinated streaming, which is enabled by coordinated bending, and you end up with this vortex that's global. This emergent axis is sensitive to details of initial data and microtubule patterning. The associated streaming flow is nearly steady, strongly vortical, fills the cell, and has speeds around 100 nanometers per second. Given that the streaming flow is essentially vortical about an extended axis, we refer to it as a twister. So this looks like a tornado, basically, where it's all sort of organized around a central point. And that central point then is sort of the center of gravity for this model. But it fills the entire cell. So it's moving around in a coordinated manner, moving things around the cell from the center of the cell out to the membrane. And so the cytoplasm has this sort of global, it's almost like a cell in the atmosphere where you have flow, you have weather that transits the Earth, and they exist within these cells at certain latitudes. And so you can see this kind of effect happening here. So there's a primary flow and a secondary flow. So the secondary flow consists of two counter-rotating toroidal vortices, one in each hemisphere, wrapped around the twistor axis. So this is like in, again, the climatological example where you have different cells for different parts of the sphere, or what is essentially hemispheres of a cell. So it doesn't necessarily need to be a sphere, but it's going to have two hemispheres where these counter-rotating toroidal vortices can occur. The simulated streaming flows can be well fit away from the boundaries as a flow with surface slip by superposing a purely 2D rotational flow with an aligned axisymmetrical bitoroidal flow that satisfies the no-slip condition. So this is just talking about the different aspects of the flow dynamics. And twistors are a combination of a strong vortical flow and a weak bitoroidal flow, as you can see here. You get these strong vortical flows here, and then bitoroidal flows, which are the hemispheres. So they do some live imaging of cytoplasmic flows. Again, here's some more modeling where they show some of the dynamics here of these different regimes of the flow. And then here's where I think they do some live imaging here. So they're actually showing the orientation in the oocyte. Once again, returning to the modeling, the structure of the streaming flow is robust. So you can see that there are these polarity vectors that occur within the cell. And you can see some of the dynamics here. So the microtubule configurations show cell geometry similar to the Drosophila oocyte from an early and a late time point. So the early time point is I, the late time point is triple I. So I is here. This is an early time point. The late time point is here. And this shows the configurations of the microtubules, kind of how they set up in both the early and the late stage. Twistors have a robust emergence where they have these different emergent states, and they can simulate this using different initial conditions. This showed self-organizing streaming to be very robust, with the main variation being the orientation of the twistor of the cell. So taken together, their results underscore the robustness of cytoplasmic streaming that emerges from hydrodynamic interactions among cortically anchored microtubules. Fine-tuning is not required. You get a lot of robust dynamics from this basic type of organization. Self-organization takes care of organizing these cell-spanning twistors, so you get both global transport and this local secondary activity. In the actual details of how the physics, the mechanics of how this work are secondary to sort of the boundary conditions and the dynamics that emerge from the initial condition. Naming such vortices twistors begs comparisons with the more familiar kind of twistor, a tornado. Tornadoes are inertial dominated and have highly localized vortical cores, maintain an axial swirl, away from which flow velocities decay. So from the center of the tornado, you get these highly stable vortices, but then as you move outward from the center, the vortex decays. Our zero Reynolds number twistors have velocities rising from the center, a la solid body rotation, and reflect a precise balance between active surface driving and viscous dissipation. So they resemble tornadoes up to a point. Self-organized boundary-driven vortices have arisen in other active matter systems, such as confined suspensions of swimming bacteria. So that's citation 38. And so this is not just something you see in oocytes, you see this in bacterial colonies and how they're suspended in confined spaces. So this is, something, again, self-organization can kind of dictate if you give it the right boundary conditions and the right initial conditions. Our model makes several interesting predictions. First of all, our twister states are volumetric structures, which predict that standard microscopy images should show a variety of flows depending on how the image volume intersects with the flow structure. So if you can look into a cell at a certain angle, or look in, in terms of where you are in the volume and where you are in the flow structure, that can give you the right information. This is consistent with our own experimental observations. Further, our simulations are statistically uniform microtubule beds and axisymmetrical cell shapes. Thus, our twisters have no preferred direction of rotation with respect to the axes of cell symmetry. So they don't necessarily match up with the sort of the structures of the cell. They just match up with how the microtubules set up the flow. Our experimental reconstructions showed no evidence for rotational biases, clockwise or counterclockwise, with respect to the anterior-posterior cell axis. And so this is not something that is coordinated by the symmetrical structures of the cell or the orientations of the cell. It's just simply something that's set up by the microtubules and the flow. Our simulations show strong secondary streaming flows originating from defect centers. So if there are defects in the layer of microtubules, or there are changes in the shape of the cell that are defective in that sense, then that can affect the streaming flow. So although rotational flow seems very robust, the secondary flows may not be, and they may be affected by various cellular inhomogeneities. So this is, again, if you have a perfectly spherical cell, you'll have a perfectly stable secondary flow or rotational flow. But if there are variations in the cell, especially over time, you can have instabilities arise. Very interestingly, simulated twisters and oocyte shapes show a slow reorientation on the long tau t time scale towards the anterior-posterior axis. So this means that there's a reorientation towards the geometry and the symmetry of the cell over time, which is interesting. And it may make sense because, this gets aligned with the cell shape as it moves around. The dynamics will sort of gravitate towards the path of least resistance. The nature of these dynamics also indicates the existence of a manifold twister state towards which the system is rapidly attracted and on which twisters slowly move towards axis alignment, which we believe to be the state of minimum energy. So this, again, is about energy minimization over time. This picture is consistent with simulations in spheres, where elastic energy also shows rapid peaking and then decay. And of course, you can have overshoot, but when you align with the geometry of the cell, the symmetry of the cell, you can limit that overshoot. So twisters serve a number of functions. Before the onset of streaming, diffusion and motor-driven transport are the main means by which different classes of RNAs are transported and anchored to the anterior, dorsal, and posterior regions of the oocytes. So RNA transport is one function. You can actually have precocious streaming, which is where things are just carried everywhere. And you don't necessarily want that. Like I said, sometimes you want local compartmentalization, but you don't want things to be locally compartmented just by default. You want that streaming to pick things up, move them around, but not just everywhere. And so this is something that can be prevented by these the dynamics of these streams as well. The streaming flow might be used to efficiently disperse the arriving yolk throughout the yolkplasm. The idea is consistent with the fact that yolk proteins are known cargoes of kinesin 1 motors. So again, kinesin 1 is known as a transport mechanism for these yolk proteins, and so that matches what we expect. Thank you for paying attention, and I hope you learned something.